My name is Iris Bertani, and I work in the CEDRIC project here in ICGB under the supervision of the leading partner that is who is Vittorio Venturi. And before starting with the webinar of today that is held by Dr. Fuzinato and Dr. Anaclerio, allow me to say just really a few words on the CEDRIC project. The CEDRIC project is an interreg project that joins 6 partners from Austria and Italy with the idea of exploiting the microbiome biodiversity of the soil to promote sustainable agriculture and development. In the CEDRIC project, what we are doing is we are making a pilot project where we try to inoculate in the field not just 1 or 2 plant growth promoting bacteria as the normal approach, the standard approach, but we are trying to make an en masse bioinoculants that involved all the rhizosphere microbiome of a rich crop in a site, in a location where the biodiversity is not so rich. So we are also creating a platform that you are invited to visit, where there is a forum where we can exchange ideas and plan for the future, always involving the promotion of scientific knowledge exchange and the develop of new method to allow sustainability to enter more aggressively in agriculture. And we are also developing a training program and a lot of dissemination events as the webinar that we are organizing here that normally during these last months has covered different type of arguments as soil health and biodiversity, bioinoculants for agriculture, and sustainability and science communication. Here is the list of the webinars that we have already organized. We are at the last, and I hope that you enjoy them. And this last webinar is the speakers that we have invited to hold this webinar are Dr. Rebeca Fuzinatto and Dr. Anaclerio. Rebeca Fuzinatto is a biologist and has a PhD in agriculture, and she is the CEO and the founder of a company, the name is Simbiotica, that is dedicated to develop sustainable solution for agriculture. And Dr. Anaclerio is an expert in regulation for allow biofertilizer to reach the market, and he works for Expedia, that is a company for consultancy in the regulation field. So now, before leaving the floor to our speakers, just remember to shut down the microphone. Remember that the webinar is registered, and then you can have access to the registration in our CEDRIC web site. And remember, if you have questions, we have a time at the end. You raise your hand and we will give you the word. So now I leave the floor to Rebeca that will start the presentation.
All right. Thank you, Iris, for the introduction. Good morning to everybody. My name is Rebeca Fuzinatto. I'm a biologist, and I have been working as a researcher basically my whole professional life. And some years ago, I decided to open this company called Simbiotica. Briefly speaking about Simbiotica, it's an innovative startup in the agri-tech sector. It was founded in 2021 as a kind of spin-out from ICGB. At that time, I was working as a postdoc in ICGB, coordinating some projects in collaboration with companies, and we saw how great was the potential of bacterial strains to become effective fertilizers to be used in agriculture. So in that context, I decided to found the company. And what we do basically is research and development services for the development and prototyping of microbial fertilizers, focusing in bacteria with plant growth promotion. We have also collaborated with a company in Serbia called Fertico since 2029. And in some years ago, this company became a part of Simbiotica became a part of Fertico. So officially now we are part of this bigger group called AgroMarket. And although we do more consultancy and research and development services, we also want to establish and promote in the future a brand, Simbiotica, our own brand. And with this webinar, what I want is to share with you my experience in these 4 years. I believe there are many researchers watching this webinar, and as researchers, we sometimes don't pay attention to things that are important outside the experimentations. And so I will share our pipeline and some challenges that I found during this path. Basically, we follow these steps that you can see in the slide, not specifically in this order. Sometimes we have a possibility to do implant a test before having results, complete results in the laboratory. And all these steps are interconnected. And it's very important that we are aware of all these steps before starting the development, especially at the regulatory level. But I will focus in the first three phases of microbial fertilizer development, and the regulatory. My colleague Matteo will explain better because he's an expert in this field. So as any researcher, we start with a starting point. So if we want to develop a product that contains bacteria as main agent, we start first from which is the target. So if the target is very specific, like we want to develop a foliar fertilizer for maize that enhances nitrogen fixation, we took that as main goal and we plan all the protocol based in that goal. But in some cases, we also received some very, very unspecific requirements. Like we had a company once that asked our consultancy to develop a special natural fertilizer for vegetables. Very, very wide. And I asked what exactly you wanted to improve quality, you wanted to improve strength, are these vegetables getting sick easily, you want to improve nutrient, which vegetables, and it was very unspecific. So at that those cases, it's up to us to elaborate some ideas, some goals. We do a lot of study on which could be the more interesting crop, which could be the more interesting goal for that crop, and we elaborate and present that to have a more specific line to follow. And once we have this target, if we don't have the strains, we do the isolation, or we do a collaboration with some institute like ICGB. We still collaborate a lot with ICGB to do that part, or we acquire in license some strains that are already at least identified. And then we do what is one of the most important things at this path, which is the functional tests. So if I wanted to see the potential of that strain, we do some lab work to verify which is the potential. So, like phosphate solubilization or nitrogen fixation or a production of lytic enzymes and so on. In many cases, if we have the possibility, from that part, we jump directly to implant testing. In other cases, we deepen the analysis, like using advanced techniques like microbiome analysis or omics to investigate further. For example, if we want to focus in elaborating a fertilizer that contains a group of strains, we like to do compatibility tests using molecular approach because it gives us a good response, a much precise response to move forward in the next steps. And we like also to create, like for each strain that we are working, a dossier, which is a documentation that contains all information regarding that strain. So information regarding growth conditions, isolation, biochemical characteristics, tests that were done that went well, tests that were done that didn't went well, everything in one documentation that facilitates a lot for further investigations. Regarding in-plant testing, we start with controlled conditions, so in greenhouse conditions. In some cases, as I said, if we have the possibility, it already happened that with few information, we had the possibility to do directly the field test. We also do that, but following a precise line, we prefer to start with controlled conditions in a way that we can test many kinds of applications. So application in the seed, in soil, in foliar. This is important to leave more flexible our possibility of placing later that product in the market. Also, we tested a number of applications. It's great when you can do everything with one dose, but in some cases, if you apply the bacteria in specific phases of the plant development, you get a much better result. And when we do that, we are always attentive to apply this second dose together with another intervention that the farmer should do in the field in order not to give double work for him. Like I will give you an example. We recently presented a product that was for seed coating for some farmers here in Italy. And they said, but we already buy the seeds coated. So why we should do additional work? But fortunately, we had already done all tests with that same product applying in the soil. And we could suggest for them, and they were happy with this suggestion and were also willing to try the product, knowing that they can apply in the soil together with other interventions. So this is important also to have in mind. And as cell concentration, we focus in 10 to 8 to 10 to 9 colony forming units at the expiring date. So after we complete the shelf life tests, at the expiring date, the concentration should be at least 10 to 8. Regarding the field tests, once we have the results from greenhouse, we do in experimentation fields, like the scientific method, like, randomized blocks, more than one parcels per treatment, and everything very well identified, very clear experimentations. But the best field trials that we have are in agreement with farmers, because they give us a feedback that is crucial to do some adjustments in the product to understand what is the real need for that crop and for them. And at this point, I must say that as Simbiotica, I never thought that doing agreement with local farmers would be so difficult, at least for Simbiotica. Now with our mother house, they have a consolidated name in the market, so it's easier to approach farmers. But initially, in my case, when I approached some farmers with the intention to establish this kind of collaboration, I found that some are open to test new solutions and others were very skeptical. And it's okay, because it's a new company with a new solution that they maybe still don't know very much. So, of course, they require proof of effectiveness. And in one case, we had also to do an agreement like indemnization in the case of yield loss. Fortunately, it didn't happen. It was a good good result. And also farmers, they like also to rely on the cooperatives, and it's a good package for them, because the cooperatives, they give all the support necessary, like for the sales of their products, for the access to public incentives. They give technical training. So why they should use a new solution if they already have all the package well established and they are happy with that? So this was one of the challenges that I found during this 4 years of company, and now I understand how the product should arrive to them, in which condition, and to whom you should talk to establish this kind of collaboration. And the validation, and here I will not enter in details because Matteo will explain better that. But once you have, you are sure that you wanted to register that product, to commercialize that product at national level or international level, you have to perform field trials with certified agencies, like like some companies, like in the Expedia has also in the group a company that is has these good experimental practices certification. So we have to confirm the field efficacy with some certified agency. And depending on where you want to register, there are different categories. And this is one point that was also important for us to decide. And I will not enter in details. Regarding production and formulation, also here the formulation involves a lot of lab and pilot plant work. We test, like in many cases, different media and supplements in order to get a higher cell concentration. So in some projects, there are always there are also the objective of getting a higher metabolites concentration. But in our case, we focus much more in obtaining the highest microbial biomass as possible. And in formulation, we test 2 different things. One is we try many additives in order to increase the shelf life. So this is a never-ending story, because the shelf life must be as long as possible. And we have noticed that at least 1 year is necessary. It's very difficult to, even if the product is very good, if you have expiring date less than 1 year, it's very difficult to place that product later, because that means the farmer can use in only 1 season. And if they buy the product and they don't use everything, they wanted to use that leftover in the next season. But if the product expires, this is not possible. So we keep doing tests to increase the shelf life, putting different osmoregulants, stabilizers, and other additives to increase it. And then the second part of the formulation is to test the possibility of transforming in powder, granular, transforming the liquid formulation in other types of formulations, which is also important depending on the type of application. And here also I had another, let's say, challenge that I was not very aware in the beginning. That's why it's important to know very well the regulatory part before starting. I was very sure that a natural fertilizer, like a microbial fertilizer, would match perfectly with organic agriculture. Well, if in organic agriculture we wanted to use as less chemical or no chemical as possible, then these kind of fertilizers are a perfect solution. But for example, in Italy, under the national law, additives are not allowed in organic agriculture. So this is very important to have in mind, because if we are aiming in a crop that is largely used in organic agriculture, we cannot use we cannot use a biostimulant or the microbial fertilizers that contain additives, unless we don't put additives, but then that would mean to use the product fresh, and this is also very difficult with logistic and everything. So I just wanted to highlight that because it was something that I discovered, let's say, a little late during my path. And there are also other things that are to be attentive regarding the regulation. Not only this part, but also, for example, in European regulation, you are very restrictive regarding the microorganisms that you can use or the formulation that you can use. While at national level, there's not this restrictive demand in the microorganisms, but there are other demands that could make you readapt all your formulation, all the goal that you have planned in the beginning. So this is something to pay attention from the beginning before starting the first steps of fertilizer development. Then when everything is settled, we start the production and we elaborate some quality control protocols with analyses that are demanded by law, for example, presence of pathogens or heavy metal contaminants, and our internal parameters of quality, color, size, odor. Odor, for example, working with bacteria is something that we consider in order to leave the product more pleasant for application. Okay, I will not talk about the regulatory part, but I just want to say that following this pipeline, we have developed, together with the mother house, some microbial fertilizers that are part of a broader, larger technology called speed technology. So this technology focuses in a triple action approach. We developed products to treat seed, plant, and soil in order to give the plant everything they need. And regarding the seed part, all the products of this line are biologicals, are microbial fertilizers. So very quickly talking about this technology, we have developed 4 formulations for wheat, soybean, sunflower, and corn. They are liquid for seed treatment, but as I mentioned, we test many kinds of applications. So these fertilizers, they can be also applied in the soil with quite good results. The main agent are the microorganisms, but we also use some microelements and plant extracts to enhance their effect. And for these formulations, more than 40 field trials were performed and many bacterial preparations were tested in greenhouse, in the field, and in the lab. We noticed very good effects in general, not only in plant nutrition, but also we saw some additional effects in reducing stresses. And here I will show you these 4 formulations. We noticed that the yield increase that they could provide vary from 6-8 to 12-13%, which is great. And this extra effect that I mentioned, we could observe especially in sunflower and corn. In sunflower, we saw that we were not expecting that, but there was one year that many fields were affected by a heavy windstorm. And you can see by the photo where the product was applied, the plants were much stronger. They could recover from this stress much faster than the control. The branches were more elastic, the plant was more healthy in general. So it's one confirmation of how investing in soil health, in plant health could be beneficial, especially when there is some unexpected stress. And in corn, we noticed also in one of the fields that we were experimenting, there was an infection of Fusarium. And one of these bacterial combinations was very effective in preventing the infection of Fusarium. You can see in the photo. And because of this good result, we used this formulation this combination in the final formulation. And still regarding corn, I must say that it was very useful to have done also a molecular approach, because we were focusing in giving strains a synergistic effect. And we did many microbiome analysis in the compatibility tests to see which strains could be partners in this enhancing of nutrition. And it ended up that the best combination was also very effective also for prevent pathogen infection. So this is what I have to say. Now I give the word to Matteo, and see you in the end if you have questions.
Thank you, Rebecca. Okay. Thank you. Welcome, everyone. Let's switch from the research to the regulatory part of a fertilizer development. Today, we will talk about the regulatory landscape for fertilizer products, and in particular for bioinoculants. The scientific background and research approach have already been well covered in the webinars. And in this final section, we will focus on how we can bridge the gap between scientific innovation and the development of products that can be legally placed on the European market. So after a short introduction, we will take a closer look at the European fertilizing product regulation and how it interacts with the national regulations, with some specific reference to the legal frameworks in Italy and Austria. And we will also refer to the current list of permitted microbial species already cited by Rebecca, and we will discuss the potential for expanding that list in the future. When we talk about the regulatory framework, unfortunately, we are never referring to a single regulation on just a few laws. Instead, we are dealing with a complex system made up of many regulations, guidelines, national laws, and so on, all of which need to be considered at the same time. This is especially important because many of these rules overlap or coexist. Here I have included only the most relevant ones for today's topic. As you can see in the middle of the screen, we go from Regulation 2019-1009 on fertilizing products to Regulation 1107-2009 on plant protection products. This regulation intersects with other product-specific regulations, such as those on biocides, but also with broader cross-cutting regulation that apply across different sectors, like the so-called CLP regulation on classification and labeling, the REACH regulation on chemical substances, the directive on patents, the one on waste, and so on. Let's start with the fertilizing product regulation. I will refer to as FPR. FPR is in force since July 2019, applicable since July 2022, and it harmonizes the rules for placing fertilizing products on the European market. We will see that only a few microbial groups are currently considered. So regarding biostimulants, The regulation offers a unified framework, but sometimes limits flexibility by restricting the list of allowed species. One of the main points of that regulation is that it allows CE marking, which enables free circulation across Europe. This means that a CE marked product can be marketed in all member states without the need for costly and time-consuming registration in each individual country as in the past. One of the key innovation of the regulation is that fertilizers are defined by their function and not by their content. In fact, this new concept of product function categories, PFC, has been introduced, and specifically PFC 6 is the one we are focusing on today because it covers plant biostimulants, including microbial inoculants. I know you have already talked about biostimulants in previous webinars, but let's just do a quick recap. I'm just going to read the definition of biostimulants according to the FPR. It's a product which stimulates plant nutrition processes independently of the product's nutrient content, with the sole aim of improving one or more of the following characteristics of plant or the plant rhizosphere: nutrient use efficiency, tolerance to abiotic stress, quality traits, or the availability or confined nutrients in soil or rhizosphere. Just for information, abiotic stresses are chemical, light, mechanical, osmotic, oxidative, thermal, and water stress. And keep in mind that the efficacy of a biostimulants product, according to the FPR, must be demonstrated. So unlike what happened before with efficacy trials, as mentioned by Rebecca, where the biostimulant treatment is compared to an untreated control in a simplified view of a trial. The biostimulant claim can be considered as demonstrated if there are statistical differences between treated and untreated, with a statistical confidence interval of 85% for field trials and 90% for greenhouse or laboratory trials, which are slightly more permissive conditions compared to those for a plant protection product. Now, consider that the FPR is about, I think, 135 pages long, and the green box on the left is all that we have about microorganisms. Microorganisms fall under the component material category 7, this CMC 7, and at the moment the list is very short, and only few microorganisms are included: Azotobacter, mycorrhizal fungi, Rhizobium, and Azospirillum. Despite this list being so short, it's already proving problematic because there is inconsistency in how broad or narrow are the terms that are used. For example, mycorrhizal fungi, it's a very broad term that includes hundreds of species, while Rhizobium spp is much more specific, and formally only species within the genus Rhizobium are included. So this could be a very important limit and problem. In November 2023, the European Biostimulants Industry Council published a position paper suggesting that the term Rhizobium spp should be replaced with Rhizobia. a very broader term, taking also into account that the genus Rhizobium has undergone many taxonomic reclassification over the years. The diagram on the right is taken directly from that position paper, but it's important to note that this is just a position paper. So while a significant step forward, it's not legally binding. That means that until the FPR is formally amended, there is still a risk of non-compliance when using a microbial inoculant with species that are not explicitly listed in the regulation. There is also a European standard that provides standardized method for counting rhizobia in biostimulants that support the same proposal. Since we are talking about soil inoculation, just a fun fact, that even though the definition of soil improver includes improving the biological activity of the soil, under the new regulation, a soil inoculant cannot be classified as a soil improver. That's because, as of now, CMC7 of microorganisms can only be used in microbial biostimulants and not in other product function categories. It's a purely formal or lexical distinction, but it can cause some confusion, in my opinion. Let's take a quick step back to better understand what CE marking actually means. It applies to a wide range of products, not just fertilizing products, but also, for example, toys or medical devices, sunglasses, and many, many others. When we talk about registering a biostimulant, although registering is not technically the correct term because there is not a formal registration process. What we are really referring to is a conformity assessment procedure. This procedure varies from a simple self-certification as of Module A to a tricky quality assurance of Module D1. For biostimulants, the procedure to follow is Module B plus C, that in practice is a technical dossier that must be evaluated by authorities. This process involves a notified body that is responsible for reviewing and approving the documentation provided by the manufacturer, including the efficacy studies which we spoke about before. On one hand, this requirement adds time and costs to the process, but on the other hand, it also provides a level of protection or, let me say, assurance. I mean, if a microbial inoculant, for example, including the specific species used, is approved by a notified body, the manufacturer gains a certain degree of legal and regulatory confidence. This doesn't mean that one can use microorganisms that are not in the official list. That's still not allowed. But maybe it could be applicable to the Rhizobium versus Rhizobia issue we discussed. Is the European regulation the only possible path to follow? Actually, no. The entry into force of the regulation did not repeal pre-existing national laws. So depending on what is more convenient or what is the objective, it's still possible to follow national procedures. although they can vary significantly from one country to another. Under the Italian legislation, specifically the Italian Decree 75 of 2010, the only product category in Italian is denominazione del tipo, is the second column. The only product category that allows the use of microorganisms is inoculo di funghi micorrizici, that is to say mycorrhizal fungi inoculum. This category appears in two separate sections of Annex 6, which deal with the so-called prodotti ad azione specifica, that is specific action products. Mycorrhizal fungi inoculum can be classified either as a soil-acting product at the top and or as a biostimulant. With only slight differences in the requirement for each. In the green square, you can see that for biostimulants, the regulation specifies a minimum concentration for both fungi and bacteria, with the specific phrase capable of forming entry points into the roots of the host plant higher than 30%, which is quite difficult to demonstrate because there is not a specific guideline or ad hoc technical specification available for Italy. So both categories are quite strict, and for example, endomycorrhizal fungi must be present only on sorghum roots. This is a very big limit, and the substrate must consist only of organic soil improver. I have to apologize for not being fully familiar with Austrian national legislation, but based on what I've been able to find and to translate, the main national references are, I won't even try to pronounce in German, sorry, is Fertilizer Act 2021 and Fertilizer Ordinance 2004. Regarding soil inoculants, Section 13 of the 2004 ordinance describe the category of plant aids and specifically mention rhizobia for inoculating Fabaceae. This is quite a broader definition than the Italian one, which only allows inoculation of sorghum, that is a grass species. The Austrian regulation also specifies some minimum requirement for rhizobium, and it's interesting that it doesn't appear to be a requirement to use an organic soil improver as a carrier, as a mandatory carrier, unlike in Italy. So in summary, with the entry in force of European regulation, existing national regulation were not repealed. This means that in most countries, specifically those that already had national legislation on fertilizer, companies can choose whether to follow the European regulation or the national rules. Of course, there are many factors to consider when make that choice. Just a few words on mutual recognition. In some specific cases, there is an alternative regulatory strategy. That is to say, you can register a fertilizing product in a country where it is in compliance with national laws. And once that's done, you can submit a mutual recognition request under Regulation 2019-515 to another member state. This approach can help so to speak, bypass certain national barriers, allowing a product to be sold in a country where it wouldn't have been possible to register it directly. However, this procedure is not always as promising as it might seem. One of the main challenges is the length of the process. In fact, although the regulation sets a maximum of 6 months of timeline, in practice, mutual recognitions can take about 1 or also in some cases 2 years, and that is on top of the time needed to obtain the initial registration in the first country. The process is also subject to evaluation by authorities, so recognition is not guaranteed, but it's important to know that this is a possible path. Which regulation should we follow? We actually need to keep all of them in mind, but the one we must comply with mainly depends on the claimed function of the product, as well as its composition and production process, depending on the regulation. Since we don't have time to give to go into too much details, let's focus on the main functions that are relevant to our topic: fertilizing, plant protection, maybe also biocidal action. It seems easy to distinguish, but often we are dealing with the borderline or dual-use products. Yes, there is a Sanco document that offers some partial guidance on these borderline cases, but in reality, there are many products on the market that are out of this guidance, and we might even say potentially illegal. Let me give you an example of a completely fictional product, just to understand. I call it RazoGuard Plus. Let's imagine its description goes like this: a biostimulant, a bioinoculant based on a consortium of mycorrhizal fungi and plant growth promoting bacteria, enriched with copper oxide nanoparticle encapsulated in a biodegradable organic matrix. I repeat, it's fictional, but many similar products are on the market. The functions listed on the label could be, for example, improve the absorption of nutrients through mycorrhizal symbiosis, stimulates root growth and tolerance to abiotic stress, reduce the presence of fungal pathogens in the soil as Fusarium and Pythium, thanks to fungal competition and antimicrobial action of the nanoparticle. So why this case can be considered as a borderline case? The product can be considered a biostimulant or soil improver since it enhances nutrient efficiency. But if there is also a claim of action against pathogens, then the product will fall under the plant protection regulation. And if the copper nanoparticles have antimicrobial action, the product might even be classified as a biocide. So this is a typical example of a regulatory gray area where the classification of the product depends on how it is presented on the market and which effects are claimed. Let's move to a real-life example, something simpler but still tricky from a regulatory point of view. I'm talking about these alfalfa seeds that are sold already inoculated with rhizobium and mycorrhizal fungi. It's not clear which regulation should apply here. Should we refer to the legislation on seed marketing? In that case, the seeds are not supposed to contain inoculants or active substances. Or should we look at the national decree or other EU regulation? or maybe this fall under the so-called free sale of non-regulated products, or perhaps it shouldn't be marketed at all. Well, I don't have a quick answer, and that is why consulting company exists. So, what we can say about regulatory strategy, that is a topic mentioned in the title of the webinar. First of all, it's case sensitive. It depends mainly on the composition of the product and its production process, the intended function, the target market, and so on. What we can say in all cases is that it's essential to evaluate the regulatory option from the very beginning. Otherwise, the risk is ending up with a fully developed product that cannot be sold. I know that in the world of research, the goals are often different from those of product commercialization. But if you are thinking about a practical application of your research outcome, I strongly recommend assessing its regulatory compliance right at the start of the project, not at the end. And so a company, a university, a research center that developed the product is the one that has the knowledge and the vision about its potential use and future application. By working with a regulatory department which understands both the technical and legal aspects, you can assess the feasibility of the process and also estimate timelines and costs. Okay, we have reached the end of the presentation. We have looked at what the European regulation offers, compared it with national alternatives, explored the possibility of bridging between different countries' regulation, discussed the current limitation regarding microbial products. As for the next steps, all we can do is keep monitoring updates to the regulation and wait for a new list of microorganisms to be expanded. A little spoiler, a new list of microorganisms is not expected for at least another 1 or maybe 2 years. Thank you for your attention.
Thank you, Matteo. Very, very interesting. Also a bit confusing, because now the idea are not more clear than before, because it's really a mess, if I can use this term, a lot of variables, let's say.
Yes, it's quite complex.
Yes. So please, if somebody has some question for Rebeca or Matteo, it will be nice. So raise your hand. In the meantime, people think I have a question. One for Rebeca.
Sorry, Iris. I just have to say, at 11 o'clock I have to go. So I'd just like to thank Rebeca and Matteo for a very nice and interesting webinar. And I apologize, I have to leave because I have a meeting in 2 minutes. So thank you very much.
Thank you.
Thank you. Bye.
So just a brief question for Rebeca, that when you are doing your trials for see if you have an effect of biostimulants or plant growth promotion, you say that you go for a final concentration of 10 to the 8, 10 to the 9 CFU per ml. But of this suspension, microbial suspension, then if you can say to us, how much is the amount of bacteria that you put for each pot or each plant, or I don't know, kilogram of soil that you treat? What is the number that you normally use to check for bacteria?
For those formulations that we developed for maize and soybean, it's 200 ml per seed dose. So the amount of seeds that covers 1 hectare, we put 200 ml. And for wheat and sunflower, it's 100 ml.
Okay. So big numbers, not really lab measures, but I will try to make a calculation. Okay, I saw that there is wait, one in the chat. Manel. Oops. Which Manel is asking, which are the types of additives that aren't allowed when you want to commercialize a product? I don't know if additive were Rebeca or Matteo. I don't remember who was speaking about the prohibition to use additive.
I think Matteo's know better this answer.
Okay. There is a positive list of what is allowed for organic farming, and all that is not on the list is excluded. So I can remember all the additives, but some natural additives are allowed, but many commonly used additives are excluded.
And these lists are published where one person that is interested, where can find it?
Oh yes, it's I think it's a regulation, but it's a published list at the EU level.
EU level, okay.
Yes.
And then just a comment, the Cedric project uses some barley seeds as for making his experiment. And I was really surprised to find that these seeds were coated, in fact, was a problem for us in the moment, with Pseudomonas chlorophyllus, but I bought here in Italy, and they were produced in Austria. So everything fits with what you said, that they allow the use of, for example, Pseudomonas chlorophyllus. We don't allow, but in the shops you can find and you can use the products made in Austria. So very intricate metrics that you can
yes,
find.
Unfortunately, on the market there are many products that are not completely legal or completely compliant with the national legislation. The problem is that there are very few checks on this.
I think that this was I hope it's legal what we bought, that this was following about in the bridge that you say that one country can allow.
It's possible that mutual recognition
yes,
has been followed. Hopefully, yes.
Great.
I think it's difficult even for a controller to say if the product is in compliance with the national law or European law, because it's so
that's true.
So intricate situation that it's very complex.
And sorry, Matteo, perhaps I lost myself during your presentation. At a certain point, you mentioned sorghum, but I miss why you mentioned it.
Because for the Italian decree, the mycorrhizal fungi inoculum is allowed only on sorghum roots.
So just on sorghum fields, we
yes.
can apply this product. Okay.
It's possible to add new categories to the Italian decree, but it's a very long process and costs and time consuming. So it's quite stuck from many years. So to date, the mycorrhizal fungi inoculum has just this very strict category.
Okay, I didn't know. Thank you, because I was missing the point. So if no other questions are, I hope that everybody enjoyed the presentation. It was really very interesting. I hope that everybody enjoyed all the webinars that Cedric organized. You are the last for the moment, at least for the moment. And I have just to say thank you to you, to Maria that organizes every time these events, and to
Thank
everybody
you.
that has that has participated to the series of webinars. Thank you very much, and I hope we can meet again next year with another perhaps presentation. Okay.
Thank you.
Thank you.
Thank you
Thank
very
you.
much.
Thank you, everybody.
Thank you very much.
Thank you. Bye.